talk at this session will be Soufiane Mourragui from Dell. And the talk will be about the domain adaptation approach to transfer predictors of drug response from the preclinical models to tumors.
Thank you very much for this introduction. So my name is, do you hear me? Yeah. So my name is Soufiane Mourragui, and today I'm going to talk about Precise, a domain adaptation framework that we developed to transfer predictors of drug response from preclinical models to tumors. And in all this presentation, I'm going to consider two kinds of preclinical models. So the first one is cell lines that are basically human tumors grown into a petri dish. And the second one is our patient-derived xenografts, or PDX, that are human tumors grown into immunodeficient mice. And ultimately what we want to see here is the difference with the human tumors. So with the real human tumors, real human patients. The problem is that it has been shown in the literature in the last 10 years at least, that these preclinical models show a lot of differences with the real human tumors, which is understandable because they don't have any immune system or any microenvironment for the cell lines. So what we can do to compare them together is to rely on comprehensive molecular profiles. So huge screening process led by international consortia like GDSC or Novartis PDXC or TCGA for the human tumors. However, when it comes to drug response, then we are really limited by the scarcity of drug response data in the humans, where we basically only have survival data, which is really limited to, so we are really limited when we want to train a predictor, a statistical model on these human tumors. So ultimately what we want to solve here is to transfer a drug response predictors trained on these preclinical models, so either cell lines or PDXs, and try to see how it predicts on the human patient, so correct a statistical model to predict on the human patients. So for that, I use three data sets. So the first one is the GDSC 1000 in the cell lines, the Novartis PDXC on the mouse models, and the TCGA for the patients. So I used around 1,000 cell lines, 50 breast cancer cell lines, 400 PDXs, and around 1,100 breast cancer patients. In this study, I solely use transcriptomic data, so gene expression in the form of RNA-seq, and I use IC50 for 44 drugs in GDSC 1000. And IC50 is the concentration needed to kill half of the population of human tumor cells. So before even going to transfer anything, we first should see whether there is any similarity between cell lines, PDX, and human tumors, because otherwise there is not even a point to transfer anything. And for that we use the notion of cosine similarity matrix, so which is a comparison of biological factors. So we first start by processing cell lines and tumors data independently, so in two batches. We perform a simple principal component analysis on each of them independently, and we get domain-specific factors, so one for the cell lines and one for the tumors. So here I take the example of cell lines, but that's of course extendable to PDX as well. And then from these two sets of principal components, we simply compute a cosine similarity matrix, which is an inner product between these two sets of principal components. And we hope to get something that is roughly diagonal, which would say that somehow the cell line components that represent biological factors will have a one-on-one similarity to tumor components. So this will be an ideal scenario where everything is transferable and cell lines behave directly like human tumors. Fortunately, that's not what happens. So when we compare breast cell lines to breast tumors, we have this kind of plots where it's like really low cosine similarity values, off diagonal, no one-on-one similarity. So there is really something, there's no clear correspondence. And when we look at tumor variance, so what you see here is the proportion of the tumor variance projected on the tumor principal components on cell line principal components. So we take the tumor data, we project it on several components, and we look at the ratio of tumor variance. We really see that the cell line principal components barely account for a third of the tumor variance. And it's not monotonic, so that's something that we can see here. So really the correspondence is really minimal. When we go to PDXs, we see roughly the same patterns with roughly larger values for the variance and for the cosine similarity, but yeah, that's still not far away from what we would expect in an ideal scenario. So there's no clear one-to-one similarity. Of course, what we want to know as well is whether there is something that we can use for transferring, and for that we performed the permutation test. So we completely permuted the gene ordering and redid this experiment again. And what we see is that the cosine similarity values drop by 2 orders of magnitude, and so do the variance explained. So which hints at some similarity that exists between these preclinical models and the human tumors. So to sum up this first part, we show that there is a common structure but no clear correspondence. And now we're going to present the method that we developed to capture this common information that we can then use into a drug response prediction model. And for that, we use the mathematical notion of principal vectors. So I'm just illustrating it with a 3D example. So if your tumor, so if you consider 3 genes, and you consider that the 2 main sources of variation of the tumors lie in this space, in this plane, the 2 main sources of variation of the cell line lie in this space, the first set of principal vectors will be like 1 vector from the cell line and 1 vector from the tumor that are as close as possible from one another. So here it's simple because the two spaces intersect, so you have 1 shared vector. But then if you want to define a second one, you will define orthogonality to the first. So ultimately here, this will give us this direction. So basically we define 2 sets of principal vectors, so in each, so 1 for the tumor and 1 for the cell lines, that are now ordered by similarity. The top one are really geometrically similar, and the bottom one are non-similar whatsoever. So of course, this is only a 3D example, and we want to go to all the protein coding genes, so it's a bit more complex. But we can still rely on the closed form solution for this problem. So if we use the cosine similarity matrix that I exposed a couple of minutes ago, so with the inner product between the principal component of the cell lines and the principal component of the tumor, if we perform a singular value decomposition onto this matrix, so we get 2 orthogonal matrices, U and V, then basically U defines orthogonal transformation that rotates the principal component of the cell line to transform them into the principal vectors of the cell lines. And this is the same for the right matrix that rotates the principal components of the tumors in such a way that it aligns them to the cell lines principal vectors. So the first sanity check that we can do is to see how these principal vectors now compare to one another, so using the same cosine similarity matrix. And what we see is now we have really 1-to-1 similarity. Of course, the diagonal value are not 1, which will be the really ideal case, but this is already discarded because of the first part. And what we see is that between the breast cell lines and the breast tumors, the first similarity are quite high. So it ranged from 78% to 2% for the last one. And well, if 78% doesn't look really good somehow, please notice that if you go to this kind of dimension and you take two random subspaces, you will get something around 10% maximum for the top similarity. So this is actually a pretty important similarity. And when we go to the PDXs, then we see that the similarity is larger, which is somewhat expected because the mouse model mimic the human tumors better. And we see the exact same pattern. So now we have principal vectors that are corresponding to one another. So we literally align the principal components. And these principal vectors, at least the top one, the one that are at the top, they really capture the common signal. And this is this one that we want to use for further processing. So before going, what we did here, so I'm just summing up a bit what we did so far. So we took human tumors and preclinical models independently. We compute preclinical factors, so with principal component analysis. And then we aligned these principal components using this principal vector trick based on similarity. So here the first pair of principal vectors are really similar. So here you can really see that they have roughly the same loadings. And when we go down the order, then we really lose in similarity. The bottom one don't share anything at all. What we want to make sure now is whether this geometrical similarity transfers to some kind of biological understanding. And for that, we performed a gene set enrichment analysis. So we first projected the breast tumor data on cell line and tumor principal vectors that we computed on this data set. We then performed a gene set enrichment analysis on the projected values. We used for that a 1,000 sample level permutation to assess the significance. And then we selected 25 gene sets, so the 10 most enriched gene sets in the top 5 PVs, so that were enriched for both source and target principal vectors. So source are preclinical models and target are tumors. The top 10 most enriched in the bottom 5 PVs, so in the PVs that are that are having contrastive information. And in this case, they were enriched in either one or the other. And all of the time, they were only enriched in the tumors. And we took all of the gene sets that were related to the extracellular matrix. And what we see is that in the top 5 PVs, all of the processes that are enriched are related to the breast cancer subtypes and to the cell cycle related pathways. And this is really something that we would expect, because when you put a human tumor into a petri dish, you would expect it to grow and to have the same invasive behavior that it has in human tumors. So roughly speaking, these processes should be present in both of them, and we retrieve them in these top principal vectors. When we go to the bottom one, we see that these principal vectors are really highly enriched for immune-related pathways and only in the tumors, so there is no immune information in the cell lines, and for extracellular matrix, which are exactly the information that are missing in the cell lines. So ultimately, discarding the bottom PC, so the one that are the least similar, really removes completely the information that we want to get rid of because it's not present in the tumor. So here we use, the output of this algorithm so far is to have pairs of principal vectors that are highly correlated. That's really something we want to get rid of because we don't want to have 2 features that are highly correlated. So there are different methods we could, there are different methods. We could use both vectors from the same pair or one or the other, but the problem is if we only use the principal vectors from the cell lines, then we will somewhat go back to the principal component analysis with a filtering step. If we use the one from the tumor, then we lose a bit of information because ultimately we will train on cell lines. So what we decided to do is to find one in between. So here, of course, for this first vector, it's really easy because this is there is only actually one vector. But for the second pair, what we do is to perform an interpolation between the preclinical principal vector and the tumor principal vector to find one possible interesting factor that will basically balance the effect between cell line and tumor. And for that, we first project the data, so preclinical data and tumor data on each of the points in between, so we discretize it, of course. Then for each of these points, we compute the Kolmogorov-Smirnov distance between the preclinical data projected and the tumor data projected. So it gives us a score of how similar the 2 datasets are in this projection. And then we take the point where this Kolmogorov-Smirnov distance is the smallest. So this gives us a consensus representation, and we do it for all of the principal vectors pairs. So we show here that the principal vectors align to domain-specific factors, align the domain-specific factors based on the geometrical similarity. We show that this geometrical similarity corresponds to some biological intuitions. We generated, based on each of these principal vectors pairs, consensus features that balance the effect between each dataset. And now we want to use these consensus features to predict the drug response in humans. So this is why we developed Precise for this purpose. So to sum up the consensus representation, so we start back from these principal vectors that I exposed a few slides back. We perform a cutoff in similarity, so we just discard information that is irrelevant, so these principal vectors that are enriched for immune infiltration or extracellular matrix. Then we perform this interpolation and this consensus representation computation, and now we will project all of the data on these consensus factors, on these common factors, in order to train a machine learning model that will predict the drug response on cell lines and hopefully transfer on human tumors. So the first check is to know whether we are still learning something on the cell lines, because maybe we just completely discarded information that is relevant for learning predictive models. So we used a compendium of 84 drug cancer type combinations. So basically we took one drug and the tumor type that corresponds to this drug, for instance, lapatinib and breast, because we know that lapatinib has an effect in breast cancer. And all of these drugs were either cytotoxic or targeted therapies. For each of these combination, we used the RNA-seq from all of the cell lines. So we didn't use one cell line, one type in cell lines, but we used only one tumor type, for instance, breast. And then we use a baseline, that is the ridge regression, without any domain adaptation, so solely on the cell state of this the cell space of these cell lines. This has been proved to be a state of the art in drug response prediction. And the second method is, of course, precise with a regression on these consensus features. And here I'm going to present to show the predictive power that we compute as the Pearson correlation between the actual IC50 and the predicted IC50 with a tenfold cross-validation. And what we see is a slight drop in predictive performance. So here you see the predictive performance that we get on the cell lines with a ridge regression, and here the predictive performance on the cell lines using precise. So we see here a slight drop in predictive performance that is mostly due to a loss of variability. But what's most important is to know how precise behaves in human tumors, because this is ultimately what we want to do. Unfortunately, as I said earlier, we don't have any drug response data, or really scarce. So instead, we decided to go for positive control, so really clinical behavior that our algorithm needs to recapitulate. So here I will show two results. So the first one is the ERBB2 amplification that is a predictor of lapatinib response. So if a patient comes into the clinic and gets is shown to have R2 amplification, then a clinician will directly give lapatinib because it's known to be a good predictor for lapatinib sensitivity. And the second one is BRAF V600E point mutation that is a predictor of tramtinib in skin cancer. So the same here, if a patient comes into the clinic with this specific point mutation, then a practitioner will prescribe tramtinib. And we perform the same procedure as before. So we compute the principal vectors, we then compute the consensus features, we project cell line and human tumors on these consensus features, we train on the cell lines, and then we compute a predicted IC50 on the human tumors. So for ERBB2, we really see that the ERBB2 copy number aberration status that you see on the x-axis correlate with a decrease in the IC50. That is, so lower IC50 means sensitivity and higher IC50 means resistance. So we really see that that precise completely recapitulates significantly this known biomarker-drug association. And this is, of course, here computed on another data set that the precise has not seen because we only focus on gene expression here. When we look at the second biomarker, we see here that first the V600E mutation is really stratified against the wild type mutations. So this is the first control. And more interestingly, we see that precise completely discriminate with the other mutation, which is also really interesting because if you have a BRAF mutation on another locus, the precise will not give you any significant advantage, which is something that is expected somehow. And this is also quite interesting here because no mutation data is given whatsoever to the data set, to the algorithm, sorry. And we also perform this known biomarker-drug association on 8 other biomarkers, and you can check it on the paper if you want. So to conclude this presentation, we show that precise, our algorithm, captures known common factors between preclinical models and human tumors. We show that these factors are enriched for biological processes that are expected to be shared, at least in the top one. And we show how to use these these common factors to train a model that will eventually capture known biomarker-drug association that acts as positive control for drug response prediction. And with that, I'd like to thank a lot of people. I'd like to thank first Hossein for the nice ad during the MLCSB session first. He also presented a nice paper that is really complementary to ours on multi-omics integration in deep learning using deep learning methods. I'd like to thank my 3 supervisors, so Lodewijk Wessels in the Netherlands Cancer Institute, Marcel Reinders and Marco Lor in Delft, our collaborators in the VU, Marc van de Ville, the CAMDA Travel Fellowship for their for funding my presentation, and ZunMV for the funding. I also like to highlight that we have some positions available in the Netherlands Cancer Institute. So if you are interested, you can just check it on the website, or you can just talk to me, and I can give you some contacts. I'd like to thank you for your attention, and I'd like to take any questions now.
Thank you for your presentations. Yeah, okay.
Very nice talks, thank you. 2 questions. First, when you actually project the tumor as well as the cell line and look at their cosine similarity matrix and you do the SUV, what would happen if actually you don't use these two data sources, but actually you just split one data source into two and look at actually subsets? How are they similar?
Yes. That's an excellent question. So I don't have it on the slides here, but we try to do this experiment using TCGA breast cancer dataset. We split in half, so you like two cohorts, and then we use it all. And indeed, we see like high similarity. The first PVs are around 99% or 98, and and the cosine similarity matrix is of course not orthogonal. You have some noise, but we see a trend in the diagonal.
Okay. Another one, very quickly, about the last validation part. I mean, you show very nicely that the mutation, in the sense of amplification and mutation in the BRAF case, actually you could predict. Have you tried actually the negative control, namely that if you find a immune regulator, immune modulator drug, actually your model should be bound to fail? I'm worried about false positives.
Yes, that's a really good question. Well, so we of course, so there is a limitation to our approach, which is that we use cell lines, and like for instance, immunotherapy is not, well, you cannot really screen cell lines easily with this kind of new therapeutic strategies. But no, we didn't use any negative control. That would be interesting actually to see, like for instance, failed clinical trial, how they would. Whether we could have predicted it with using our method. Yes, that's something that we want to investigate further. Yes, indeed.
Hi. Interesting presentation. Thanks. I like this idea of these principal vectors. I wonder whether there's any relation to canonical correlation analysis, which sort of tries to do the same thing.
Yes, indeed. This is so these two notions, so the principal vectors and the CCA, share this notion of canonical angle. However, in CCA, you need to have some pairing because you need to compute this cross correlation matrix in order to find the direction of common of shared variation of a correlation. Here we can't do that because we don't know which cell line correspond to which human tumors. So we need to instead of finding direction that are correlating to one another, we first find direction that are of important variation. And then we align them together to see which of these directions are shared. So this is, but yes, indeed, this is there are some fundamental similarities between these two approaches. Yes, indeed.
Hi, very interesting talk. Thank you. I was wondering, you showed analysis transferring from cell lines to patients. Did you look at transferring from cell lines to PDXs?
Yes. Yes, we did. So I didn't put the results here. So we tried from one to the other. Yeah, so we don't have all of the results yet. For specific drugs, it works quite nicely. For instance, for trastuzumab and nivolumab, I think it works really nicely. For other drugs, we don't see much improvement so far. No, using this method at least. No. So yeah, it's the results are mixed on the cell line to PDXs.
Any further questions? No? Then thank you again.
Thank you.
And we'll